I'm Julie Phillippi, and I am faculty at the University of Pittsburgh in Pittsburgh, Pennsylvania. And my primary academic appointment is in the School of Medicine and the Department of Cardiothoracic Surgery. And I also hold a secondary appointment in the Swanson School of Engineering, Department of Bioengineering, and I'm a member faculty within the McGowan Institute for Regenerative Medicine. And my lab focuses on the role of a microvascular network known as the vasa vasorum and how it influences aortic biology and other great vessel disease. So we are really interested in understanding how these microvessels relate to their environment of the extracellular matrix and how we can harness their potential for regeneration to develop less invasive and preventative treatments for patients with cardiovascular disease.
What are the ongoing projects that you're currently focusing on in your lab?
We have several projects that span understanding these microvessels. Most of our work has been in the space of aortic biology and understanding their role in aneurysm formation and progression. We also have some basic biology projects where we're looking at the relationship between biophysical cues of the extracellular matrix and biochemical cues and how they influence cells associated with the vasa vasorum, such as endothelial cells, and in particular, a big emphasis on pericytes that surround these microvessels. We also have some smaller projects that are related to lung failure and the role of vasa vasorum in lung failure. And for this, we're looking at patients who are undergoing lung transplantation. And we have a few collaborative projects in transplant biology that are new and coming down the pipeline.
What is something that is exciting to you about this line of work?
I feel energized in working with people from different training and different backgrounds. We partner very heavily with cardiothoracic surgeons and rely on their expertise to help guide the experimental design, also provide access to essential specimens from surgical procedures. So, for example, for our work in aneurysm biology, we bank the tissue that's resected during aortic replacement operations and aortic valve replacement, also during heart transplantation and lung transplantation. So we have a tremendous resource at the University of Pittsburgh and the University of Pittsburgh Medical Center, a very large and robust, prospectively collected tissue bank of vascular specimens and cells. So we're able to do histology, live cell work, lots of tissue culture model work, and tissue engineering approaches to ask our research questions. So I think it's really exciting to be close to patient care. We consider our lab to be an extension of the operating room and interface with the clinicians, surgeons, and other health professionals to accomplish our goals. So I'm really propelled by that, and I think it's an excellent training environment for students and fellows, also training surgeon scientists. So I think it's the present day of biomedical research and of course the future to be able to work and interact within these types of interdisciplinary teams.
What are your perspectives around mentoring and your role as a mentor? What is your philosophy around that?
Yes. So having the benefit of excellent mentors myself over the years, I think that I've sort of crafted a style that's tailored to each person's individualized needs. So basically, hands on as much or as little as needed. It's different, really, for everyone that's come through our team. And I like to be available and provide guidance and advice on projects. One of my favorite things to do is design experiments with mentees and really just be able to help them grow as a scientist, all with the intention of developing various levels of independence. I really think that all levels and skill sets are needed for a successful team. We also practice and embrace a shared leadership perspective. That's something that I was trained in, and having been a part of multi PI teams and highly collaborative teams for the entirety of my faculty career, as well as my graduate training, working within centers and institutes. And in a highly interdisciplinary space of tissue engineering and regenerative medicine initially, I think it's really important that everyone has a role and an opportunity for leadership. And the mentoring style really is curated based on the needs of the individual.
What would you say to someone that is interested in joining your lab and working with you?
So we have a couple of new and existing projects that we're looking to repopulate our team at the postdoctoral fellow level. Uh, Maybe I should also describe what the team looks like today. So we have two staff research specialists that have been working with our team for multiple years on these projects. There's a postdoctoral fellow who is a surgery resident, spending 3 years in the lab for research training. We have a visiting medical student, and we have had 4 undergraduates this semester, and one of which is graduating tomorrow, and one first-year bioengineering doctoral student. So we have a variety of team members at different levels, different expertise, different career pathways, and we're looking to add to our team both on the graduate student side as well as the postdoc side. And in particular, they would be have the opportunity to participate in projects that incorporate tissue engineered models of cardiovascular disease. So one, which is a new NIH R01 grant that's going to start in the next month or so, is focusing on pericyte biology and the role that biophysical cues from the matrix play on the pericyte's ability to migrate, to participate in and contribute to angiogenesis. It's a combination of in vitro and in vivo models. There is some mouse work involved, lots of cell biology, and lots of collaborative opportunity. This is a multi-PI grant with a colleague and friend from Virginia Tech who provides the expertise in scaffold design and some really interesting biophysical analyses to help us understand the pericyte biology. So that's one near-term project that we're excited about, and we'd like additional help with that. The second project focuses on aortic biology, the role of hypoxia in smooth muscle cell behavior, and looking at extracellular matrix remodeling. So for this project, we have several components, one of which is a clinical trial that works on identifying hypoxia in patients' resected tissues. And then we have also a vascular graft model that incorporates a perfusion bioreactor system for dynamic cell culturing and also leverages quite a bit of work with patient cells to understand the role of hypoxia and oxidative stress in aortic wall biology. So that also is coupled with computational modeling through collaborations in bioengineering, where we're pulling together all of these different metrics from tissue and cell-based bioassays with clinical imaging to identify patients at risk for aortic dissection.
How do you think your work in your lab can impact the field that you're in?
So I like to describe our work as having 2 primary goals. The first is to understand the underlying mechanisms that are driving cardiovascular disease, for example, aneurysm, and understand that at the cell and tissue level. So we're really hoping to uncover these new mechanisms to better assess who needs the surgical intervention, who needs the operation. So the reason for that is patients with aneurysm, the clinical decision making is guided, the foresight is limited, and it's guided by measurements and clinical imaging. But patients often have catastrophic dissections with much lower measurements. So that tells us we really need to understand more about underlying biology. So that's goal 1. Goal 2 is to develop less invasive surgical approaches. So for aortic replacement, it's an open heart procedure, a sternotomy is required. But what if we could deliver a biomaterial that could be delivered through a smaller incision, or something that's preventative if they're already having a bypass operation or a valve procedure or something that could be delivered robotically, minimally invasive. So that's our second goal, is to develop something that's preventative and less invasive. And that's where we see our impact in the future.
What do you think are the main challenges of this type of research?
The challenge is really, I think, in the path to translation. So we're learning so much utilizing human cells and tissues. A challenge is in developing an appropriate animal model to understand these disease pathways, and then an appropriate model to test a therapeutic. So, for example, mice have been great. They've taught us a lot about genetics, different genes that lead to different disorders, but they imperfectly represent the human. And however, we have lots of strains on resources and costs to conduct these important translational preclinical studies. So I think that's one particular challenge that we need to navigate. One way we're doing that is to develop not necessarily genetic models, but perturb other pathways that we are identifying in the human samples and recreate that in an animal model, such as the rabbit or something a little bit larger that is closer to human anatomy, to understand these disease mechanisms and then develop new treatments. So that's something that we're working on, and thinking about coupling that with the human specimen work that we do. That's one unique challenge. One I think that's ongoing is creating and managing these interdisciplinary teams. So I think it's really important for junior scientists to understand how to speak the languages of their colleagues. And your friends today will become your colleagues and collaborators in the future. So knowing how to ask the right questions of one another is really important. So it's an ongoing but addressable challenge, I think, for our field.
What first drew you into science? When did you first start to become interested in doing science as a career?
Yes, so I very clearly remember just kind of falling in love with research during high school in my biology course in 11th grade. And I had a really wonderful teacher who was very encouraging and supportive, and he recognized that I seemed to like what I was doing. I really enjoyed writing the lab reports and coming up with the new questions. We were required to come up with 5 new questions at the end of every lab report. And that was my favorite part. And I must have told him that, or he saw that I wrote 10 questions instead of 5. And he would make me aware of other opportunities locally. There were lectures at other schools or local colleges that he would let me know about. He told me that he saw me running my own lab someday, and I just never forgot that. So it really motivated me to continue. And I went to Penn State. I did a summer pre-college program through the Howard Hughes Institute at Penn State. And my major was microbiology with a minor in biochemistry and molecular biology. At that time, I thought I was going to study infectious disease. But as time went on, I had a couple internships in clinical labs. I spent 8 months at the NIH on a co-op. And I really understood that I wanted to do applied translational work. I didn't really know what that looked like or meant yet, but I wanted to do more than basic science. So deciding to go to graduate school was also encouragement from my mentors from my NIH co-op internship. And they gave me some advice that was useful to me, that if you think you want to do it someday, go do it right away, because you never know what life is going to throw at you, and it could become more challenging later in life. So I decided to do that, came home to Pittsburgh to go to Carnegie Mellon. And at the time, there was this great energy and interest in building tissue engineering teams for various organ systems and tissues. And I was really excited by that. I found the people working on that at Carnegie Mellon. They were assembling teams and centers, and there were grants and companies and just a lot of excitement around that topic. And I chose a project and a mentor I had a couple mentors for my PhD. So I had multiple projects and side projects, which was great. All centering around stem cell biology and how the extracellular matrix influences stem cells to become different lineages. There were aspects of bioprinting in the early phases. There was some scaffold-based work, but all thinking about cell and matrix signaling. So had training in that area. I continued in a postdoc in tissue engineering, kind of expanding on my PhD work. And I knew that I wanted to stay in Pittsburgh, build my career in Pittsburgh, and really academia just made sense. I really enjoyed writing papers and writing proposals. Maybe that's not what everybody loves to do, but I really enjoy it. Again, it comes back to the experimental design and writing, and that's something that I love to do. So I began looking for faculty positions in Pittsburgh, and I happened upon one that was to build a new research program in cardiovascular biology at the University of Pittsburgh and partner with a surgeon who was new to the area as well. So that's really where I switched gears from orthopedics and stem cell biology to cardiovascular biology and academic surgery research. And that was almost 16 years ago, and it's been wonderful being able to work alongside surgeons in the lab and develop some of these interesting experimental designs to address real diseases, real problems, and hopefully develop solutions.
Any advice for someone that might be interested in pursuing an academic career, but might be discouraged by all the stages and all the hurdles in the career path?
Yes. So to your first question, I think it's important to have a unique skill set and continue to hone and develop that and recognize who and what circumstances that's valued. And that helps you create your brand or your niche, I think. But aside from that, I want to emphasize the importance of partnership with collaborators, mentors at different levels. So that's something that's really been important to my career development and to fulfillment of what I'm doing. So I think it's important to build and maintain and nurture the trust that you have in your collaborators and research partners and foster that among your team.
Okay. So you enjoy that kind of collective creative thinking process that academia offers?
Yes, you have that freedom. That's something else that's really important to promote and I think model is freedom and latitude to pursue ideas. And I think to those that maybe feel discouraged by the world of academia, it does provide a lot of flexibility. There's room for different lifestyles, I think, and other interests. You can, yes, it's a lot of work. It's, the downside is the stress of pursuing grant funding. I think you're going to have stressors similar to that in other fields. Of course, my perspective is narrow in that sense, is that most if not all of my career has been in academia. I think it's something that works with the kind of life I want to have.
That's good. So one other thing, and this is mainly because of personal interest, but I saw that you have worked as an associate editor for Science Advances. So I was wondering what this role entails and how you got involved with that. And yeah.
Sure. So I think this is a great example of just keeping your eyes open for opportunities and open doors. And, it's not infrequent that we get emails about a visiting guest speaker or, somebody that's a guest to the university. And the person that's coordinating their itinerary is always looking to populate that with different meetings and have that person interact with people on campus. So in this situation, we had another editor from one of the other science family journals visiting and giving a talk for the department. And I had an opportunity to meet with her. And so we were just sitting and chatting very casually. And I was just telling her, like I'm telling you about, I like writing and reviewing manuscripts and papers and different things. And she said, well, we're starting a new journal and we're looking for editors. Are you interested? And it was really as simple as that. I sent her my CV and she forwarded on, and I had an offer at the initial stages of that new journal. So it was really exciting. I've enjoyed it. I've been a part of the journal since it launched in 2014. So it's really great way to see the latest and greatest in the field and be a part of the decision making of the science that gets published. So I've really enjoyed that role, made a lot of new connections, and has encouraged me to stretch my abilities a little bit, because we get papers that are not necessarily specific to our field, but it helps us to branch out a little bit and learn more as well. So I think what it really entails is managing papers, finding reviewers, and evaluating the reviewers' comments, and interfacing with the authors, and making decisions and recommendations to next level up, which would be deputy editors of each group within the journal, and working with them. It does get interesting. Sometimes we have to manage manuscript appeals or disgruntled authors and different things. So seeing that side of the publishing world has been really interesting as well.
How do you think being in this role has impacted the way you do or think about science?
Yeah, I think it's important to communicate science effectively, understanding your audience. So that's something that we think about as editors: is, will this appeal to the audience of this journal? The impact is a big factor in the evaluation process, and really the quality methodology, and how robust the experimentation is. So it's really helped me, I think, to hone our experimental approaches as well, and learning how to effectively communicate and appropriately communicate our results.
We're almost at the end of our time. I just want to make sure that I give you the opportunity to say anything that you thought I missed, or just anything you want to share with people.
Sure. So I think we have a really dynamic and passionate group of researchers. We'd love to recruit new individuals to join our team. I think that we also are very interested in having different layers of diversity on our team, because we believe that creates the strongest team. So we do recognize, not everybody has had the same resources or opportunities before we're able to work with them, but we do think that there's room for everyone, and we embrace that multiple backgrounds and expertise are really important for the best team possible.
Awesome. Well, thank you so much. It was a pleasure to meet you and talk to you.
Thank you.